Very good afternoon, everyone. So, is the presentation? First of all, I want to make sure that the presentation is visible as well as my voice is audible. You can put the message in the chat box. Fine. So, very good afternoon, everyone. And on the behalf of Make Intern and Learn to Upgrade, I am Dr. Ankita Singh, and today I will be taking your session on the biotechnology century, the industrial biotechnology and its workforce, different applications of biotechnology, as well as the different fields in which you can pursue your careers. So thank you so much, Makinton, for giving me this opportunity to connect with the wonderful audience so that I can impart the knowledge that I'm having about biotechnology. So my dear students, this current era is, we have so much competition in this current time. And this biotechnology, it has emerged as one of the very important field in which you can pursue your careers, your dream. But right from the very beginning of this biotechnology, this field has a very, applications are very much wide, but at the same time the scope and the we can say the promotion of this biotechnology word, this scientific and very fancy word, is less in comparison to the other areas. But if you are very much upgraded, as well as if you have a base from right from the basic, or we can say right from the beginning, knowledge about this particular thing, you can pursue your careers and you can definitely earn, as well as you can have so many startups, because each and every field, either it's the medical field, or it is your biomedical field, or it's the agriculture field, or it is the pharmaceutical field, everywhere there are so many applications of this biotechnology. So in this particular presentation, we will be learning about the basics of biotechnology, what are the different types of biotechnological field, and in the current 21st century, what is the biological challenges that we are facing, and in the future, what for different challenges we will be coming across, and how we can fight back with these challenges, and the biotechnology workforce. So basically, because you must all have studied about biotechnology, very basic definition, you must be knowing that it is using the living organisms or the products for the living organism for the sake of human benefits to make a product or to solve a problem. Now, there comes a thing that you are using the living organisms, or you can say we are not only using the living organisms that are like there are so many fancy things that you will be knowing about, that we are going for the recombinant DNA technology, we are going for the gene cloning, we are going for the vaccinations and all. But right from the very basic, if you you must have listened, or you must have heard, or your grandparents or your grandparents must have told you, or even your parents must have told you about so many basic things, which actually are the inventions of this biotechnology. But at that time, at the previous times, they were not knowing that this particular thing is of the biotechnology field or the medical field, because in our day-to-day life, this biotechnology, because this industrial biotechnology is a term that we have given now, because now we know the applications, or we can say now we have segregated the applications of the biotechnology for the various fields. But in the previous times, and years back, even 10 years back or 20 years back, there was not that much scope, or people were not knowing that much about biotechnology. It was not a very well-known term that was used. So you must have heard that how you are making the curd by using the bacteria. So it was very easy that if you have to make a curd, what you have to do, you can take a spoon of curd, or you can take a spoon of yogurt and mix it with the milk, and after 2 to 3 days you will be getting fresh homemade curd. So that was the technique. And if we talk about your pickles, that is a very common practice that is being carried out in so many villages and so many houses. They are making the pickles at home. So all the or we can say vinegar, apple cider vinegar and the normal vinegars. So these are all the things that we are doing since ages. But now when we have this specific term, industrial biotechnology, now these specific things have a special place. And obviously when these special things have a special place, then there are so many career opportunities. So you must have heard about the fermentation, selective breeding, use of antibiotics. Fine. So these all are, they have came from our ancestors. They have came from our families only. Fine. So next, you can see here, you can see two type of fishes. So, if I give the example of selective breeding. So selective breeding is also, industrial part, or we can say this is also a type of industrial biotechnology. The selective breeding is a field, or we can say this is a application of industrial biotechnology, right? So you can see here, this is a normal zebrafish. Now, this is the Casper zebrafish made by selective breeding. So this is now technology. So in the, if you, I guess most of you belong to South and North Indias, so you must be well, known about the mythologies that we all are having. So in the ancient times, many of you are, just praying to God Ganesha, and so many gods are there in India. So we have different religious cultures and all, we are used to pray. So at the time when we see Lord Ganesha, you must have seen that he has a head of elephant and a body of a human. So at that time, that was a kind of breeding, or we can say that was a time of type of use of this biotechnology. But that was the ancient time, and that is mainly we call it a mythology. We say that maybe it is true or not true, but definitely that particular thing is something that can be connected with the present technologies. Because in this picture, this is a normal zebrafish. Now what we are doing with the use of this breeding by the selective breeding, because some breeds of fishes, right, they are not that much good for the some consumption purposes, or for the improved protein content or the improved antioxidant content. What the scientists are doing, they are just cross breeding the different types of breed of fishes. And after genetically, or we can say genetically breeding the different types of fishes or different types of species of fishes, they can make a new breed that is basically prepared by selecting up two types or three types of different breeds. So that is the biotechnology. And now what that is why that is very much been beneficial, because in the market, when you're going to sell the fishes that are having hybrid, or they are specifically selective breeds, so people will prefer to purchase those selective breeds which are actually giving them high dose of antioxidant, or they are they have a good life cycle, or they are laying a good amount of eggs. So that is the application. And definitely when the scientists are working in the laboratories day and night for these all these things, that is definitely for the people, that is definitely for the people to increase the market value, because ultimately everything is connected for earning, right? So that is one of the very good example of selecting breeding. And not only the fishes, there are hens, there are goats that are crossbreed, and they are crossbreeded, and the specific good breeds are selected for the goats or the hens, and the hybrid breeds are presented into the markets, and people prefer to purchase, because there is a good amount of community that is also have this consumption of, they are non-vegetarians, and they are consuming these hens. And for the market purpose, if we talk about, then obviously if someone is selling the good breed of these fishes or the hens or the goat, there are so many people who will be purchasing because they will prefer the breed that is giving them a good quality of nutrients, right? If the egg is being sold in the market, then obviously if the egg is from a selective breeded this hens, they will prefer those eggs in comparison to the normal eggs that are laid by the hen. So in this way, the selective breeding is a very good branch of biotechnology where career can be pursued, as well as it is one of the good invention, or we can say thing that is introduced by the scientist in the field of the industrial biotechnology. So basically this feature of Casper that has made it a model organism, fine, basically to study the migration of cancer cells compared to the wild type fishes. So not only the market value or for the purchasing purpose, even to conquer many of the diseases, the hybrid or the selective breeding is done. Even in the plants or any type of animal, the cross breedings are done specifically to study the different types of cancers. For example, there is a fish that is much more prone to the cancer because of its genetic buildup, or due to the different cells or different any of the ancestry that particular fish is having. Then scientists can crossbreed and select the different breeds to make a new model organism that can have a less prevalence of the cancer, so that they can safeguard the particular breed. So also for the protection of the breed, or to conquer the different types of diseases, the selective breeding is done amongst the various animals, and mainly, or we can say specifically, the fishes, the hens, the goats are one of the major animal breeds that are crossbreeded, or the selective breeding is done, right? So when we talk about this technology and what does it mean to us, based on this tree, you can see this tree is here, right? This is a pictorial presentation of the tree. And this can tell you that how much you can be successful in this industrial biotech. And this biology and this technology, fine, it has we have just merged the two terms, biology and the technology, to make up this new field, that is the biotechnology. So you can see, based on this tree, can you become successful in the biotech industry only studying biology? That means if you are studying the biology with the aid of the technology, there's a question that you can really become successful. So you can see, this is the branches, this is a tree, these are the roots that you have studied in your this 10th, 11th, 12th, 7th, 8th, all these schooling. Fine, you can see here, these are the roots, your human and animal plant physiology, your mathematics, your molecular cell biology, your immunology, statistics, microbiology, biochemistry, genetics, physics. These are all the roots. Now this biotechnology has emerged out of these basic biology and these basic life sciences. Now when you see the stem and the branches, you can see the genetic engineering through molecular biology and analysis through bioinformatics. So this bioinformatics, this has came from the computer science, this has came from the physics, this has came from this molecular biology and cell biology, this has also come from the mathematics, right? This genetic engineering, definitely the molecular cell biology, your immunology, your genetic subject. So it is the combination of all these fields. Then when we talk about the agriculture biotechnology, definitely the transgenic crops. So when we study the plants, if you are well known to the plant physiology, you can work well into the field of transgenic crops, because just like we are talking about the different types of model organisms, that we are cross breeding or we are doing the selective breeding, so that we can at least give people the disease free animals, or the best quality eggs, or best quality meat, or best quality animal for this agricultural purpose. So that is one application. As this, in the same way, when we talk about the crops, there are so many transgenic crops, right? There are so many disease-free crops that are made with the help of this transgenic recombinant DNA technology, plant breeding, gene breeding, or the selective breeding, where if a crop is, for example, if there is a crop, there is a wheat crop, right? Wheat crop or a sugarcane crop, or we can say rice crop, and that is, more prevalent any of the bacterial disease, any of the plant pathogen, and that particular crop is prone to any type of particular pathogen, right? So in that case, what can happen? We can, produce various types of different breeds of the crop, which are less prevalent to any type of disease, and we can crossbreed them, and we can prepare a hybrid plant, or we can prepare a, large number of hybrid seeds. And when those hybrid seeds will be sown into the fields, they will be giving a good quality of crops, as well as their genes will be having the capability to get rid of any type of plant pathogens easily. So definitely a farmer will be getting much benefit with these transgenic or hybrid crops in comparison to the crops which he is having, right? And particularly if he or she is facing so many problems with the various plant pathogens, definitely that hybrid or transgenic seeds or these transgenic crops will give him a better opportunity for the market and selling the crops, or for the personal consumption. So this is the role of agriculture biotechnology, and you can see this agriculture biotechnology has also emerged out from the basic root of your biology. Then your regulatory approval oversight. Now these are some of the industry. This is also an industry, this is also a field. But for coming in this field, you should be knowing the roots of the biology, the life sciences, your Food and Drug Administration, FDA, Environmental Protection Agency, EPA, US Department of Agriculture, Occupational Safety and Health Administration, OSHA, as well as your UNICEF, your World Health Organization, WHO, your ICMR, Indian Council for Medical Research, your NABL, National Accreditation Board for Laboratory Testing and Calibrations. These all are the various National Disease Control Board. There are so many government fields, various regulatory and approval oversights that require the biotechnologist. For example, if you want to pursue your career in the Food and Drug Administration, definitely you must be well versed with the food biotechnology or the food technology, or you should be knowing well about the pharmaceuticals, pharmaceutical biotechnology, the drugs, right? For the Environmental Protection Agency, you should be well known, you should be well versed with the plant physiology, animal physiology, and the geography, the environmental conditions, because that is microbiology, that is also the part of the life sciences. For agriculture, definitely your plant biotechnology is very much required. Your genetics, your molecular and cell biology, that is also very much required. So, all these fields are the fields of the industrial biotechnology. That means an industry is using the biotechnology, or the biotechnology is an application in the industry, and that is what we are calling as industrial biotechnology. It can be any industry. It can be your food industry, it can be your drug industry, it can be your agricultural industry, it can be your any type of detection or genetic industries, it can be your pharmaceutical or recombinant DNA industries, it can be your environmental and aquatic industry. Everything has that one thing, that is the biotechnology, and it has a wide usage. The only requirement is that you should be knowing about them. Then you can see in this detection, forensics, your DNA fingerprinting, disease DNA detection, in the environmental and aquatic field, the aquaculture, bioremediation, transgenic modifications, bioextractions, in the field of pharmaceuticals and healthcare, your biopharma, gene therapy, gene knockout testing, fermentation technology. You must have heard about this downstream processes and the fermentation. These are all the uses of this biotechnology in the various industries, right? So if we talk about the modern examples of this industrial biotechnology, then we have this one of the very, a very fancy and very interesting term, that is the gene cloning. Then the other term is genetic engineering. Then your one other term that is very much, interesting is recombinant DNA technology, human genome project. These are the modern examples of your industrial biotechnology. And this gene cloning and genetic engineering, recombinant DNA technology, these all are also in work. It does not mean, although this human gene cloning, that is not a, if we see from the clinical part, it is not that much, going to benefit the human. Definitely, if you are getting a clone of yourself, that is not going to benefit. But gene cloning in the various aspects, genetic engineering in the various aspects of the, in the food, mainly in the food industries, or for the production of the antibiotics, or for the production of your vaccines, in that in this particular fields, these techniques are very much, important. This recombinant DNA technology, your hybrid plants and transbreeds, these are all, the inventions of this recombinant DNA technology. Because of this technology, we are able to have this hybrid, you must have, heard about this BT bagan, BT brinjal, and your one of the sheep that was, produced by the, gene cloning. So these are all the modern examples of your biotechnology. And when we give the example of the modern technology, this recombinant DNA technology started the modern biotech as an industry. So if we talk about this in this particular century or this particular era, this recombinant DNA technology is one of the, very much emerging field of the industrial biotechnology. And, there are so many examples of these recombinant DNA technology. Development of the disease resistance plant that earlier I was discussing, the disease free plants that are made by the selective breeding, very, very important. You must have heard about that, for this late blight of potato. Once there was, a very much havoc in the country because of the late blight of potatoes. All the potatoes were having this pathogenic strains, and because of that, the farmers had to face a very huge crisis, because their all the potato farming was destroyed due to the disease. As well as there are so many diseases which are currently destroying so many crops, your sugar cane, then your this ones, these in the particular season. If we talk about June and July, we have this lychee and mango season. There are also many diseases that are related with the plant. So they just take over the plants, and then definitely there will be not that much of yield a farmer is hoping. And because India is, this farming oriented country, our ancient background is basically the farming. Because everything, even if I say, if when someone is a doctor, someone is an engineer, someone is a scientist, the ultimate thing, whatever the research or thing that he or she is doing professionally for the people, apart from that, the earning he or she is doing is for definitely for eating, for the food. And the food is coming from, from the farmers itself. So there is very much need of the disease resistant plants, because these plant pathogens, this plant pathology is also one of, the very much required field of the modern biotechnology, the industrial biotechnology. Although we have very much, very much things to talk about this biotechnology, but when we talk about this plant pathology laboratories, there are not that much laboratories present, so that at least the farmer can, go up to and look up to these plant pathology labs and at least diagnose that their plants are disease free or not, so that they can take any actions. So definitely this is also very important. This plant pathology field is very much important. And with the help of this biotechnology, this industrial biotechnology, we can have the development of the disease resistant plants, then the food crops that produce greater yields. Definitely. To increase the nutritional value, to increase the yield of the crops, these technologies, these selective breeding, hybriding, or this RDT, recombinant technology is used. Then you must also have heard about this golden rice engineered to be more nutritious. That was one of the achievement in the field of getting the good food crops, the nutritious food crops with the help of the biotechnology. Then genetically engineered bacteria that can degrade the environmental pollutants. This genetically engineered bacteria, this is also a very much good application of biotechnology in the industry, because there are so many pollutants every day in all of, if we talk about whole this world, we talk about whole world and the different areas which lie near the sea coast, or because most of the this transportation of the oil is being done with the help of the ships, mostly by the ships. So there is a large amount of oil spills in the oceans, in the seas, and because of this, these oil layer, they cover up the upper area of the upper surface of the oceans. And because of that, what happens is the oxygen is not able to penetrate, or we can say the sunlight is not able to penetrate inside the ocean. And because of that, the creatures living around those particular areas, they don't get enough of oxygen, because these plants, these aquatic plants, they also need sunlight for the photosynthesis. But if there is a layer of oil, because that oil and water, these are both the immiscible liquids. So definitely if there is a oil layer and the water layer, the sunlight cannot penetrate inside this oil layer. And because these petrols and diesels, they are very much, they have a very good density. So because of that, the oxygen scarcity is there. Because of that, the plants cannot breathe. And plants, if plant will not breathe, the plant will not do any photosynthesis, then definitely the oxygen will not be produced. And yes, because of that, it can lead to the death of many. Fine. So it can look like it can do so many things into the particular oceans or the seas. So these are the definitely good examples that can be done in the field of this biotechnology, that you all can pursue the different careers in all those fields. So although there is a bit of there is a lack of so much communications in this biotechnology field, there is not that much of there is not that much of promotions. But yes, if you have took a step to at least do the specific things in this particular field, you can definitely go up to creating a very good career into the field of this modern biotechnology, and specifically in all these fields that I am discussing. So now, so many more examples that I will be discussing, and definitely when we will be having this session, we will be discussing that in which field you want to make your careers and what you know about biotechnology. So it will be definitely a good open platform for you to discuss that, how much aware you are about this field of biotechnology, and what are the various careers that you want to make into the field of biotechnology. So we will be open for so many discussions. So basically, you can see in this picture, this basically the descriptions of the chromosomes. Fine, you can see chromosome 13, chromosome 21. So you can see now this is a basically a session. This particular side includes a question. So you have to look at these two chromosomes, and you have to determine which chromosome has more than one gene involved in promoting breast cancer. Now if there you can open up your mic and you can take a bit of time to see this picture that is on your screen, and you can see these are two chromosomes. You can see these are chromosome 13, and this has about 114 million bases, and this is chromosome 21, and this is your 15 million bases. Fine. So you can see there are different blue marks up there, and these are all the various million bases, and they are defining the various types of factor: your cholesterol lowering factor, cataract, journal pulverulence, stem cell leukemia, x-ray sensitivity, lung cancer. Because chromosomes and your alleles, your DNA, everything is in your gene. So anyone who can explain this particular slide, that at least it's not like we will be judging you, it's just like that you can just open up your mics and speak out that what you think about that, which chromosome will be having, which can aid in promoting the breast cancer. Any one of you, or I will, just take out the name of the participant and you can speak out the answers. So any one of you, you can also put up this in the chat box. If it's not necessary to open up your mic, you can put also in the chat box. So there are so many students: Achal, Ayushi, Amandeep Kaur, then Dinesh, Dimple, Harika. So you can also write up in the chat box, my dear students, that amongst this both. Okay, fine. You can just explain what is a chromosome, because this is relevant to the biotechnology. You can explain up in the chat box itself that what is a chromosome. Then we will be now discussing about this particular chromosome, which is more relevant for the breast cancer. Hema and Harika. Okay, there are so many students. Divya R, can you write up in the chat box that what do you understand by chromosome? Or you can unmute your mic and speak out. Khushbu, Satish. Okay, there are answers up there. Fine. Transfer of genetic characteristics, right? Sadiq has answered that it is responsible for the transfer of genetic characteristics. Yes, it is there. Chromosome is a long DNA molecule with a part or all of the genetic materials of an organism. Okay, good. Okay, fine. That is a good answer. That definitely. But my dear students, you cannot write up like that long DNA molecule. You can say, chromosome is a set of different or millions of base pairs that are, constituted in a DNA molecule, which can be responsible for the various and different genetic traits or characteristics. And that particular characteristics or traits are, they are in the hierarchy, and they can transfer from the parent to the infant. Okay, Satish has answered that chromosomes are thread-like structures found in nucleus of every cell. Okay, that is a good answer. Fine, they carry the genetic informations. Good, good. Okay, student, that means you are, well versed with this biotechnology and chromosome. Fine. So next, we will go up to the question. As you can see on the slide, there are two different chromosomes. So you can answer that which of the gene is most responsible for promoting the breast cancer. Any one of you? The students who have answered: Satish, Khushbu, Arthi. Which gene is more involved in promoting the breast cancers? Can you answer? Okay, there's one. Okay. Khushbu thinks that it is chromosome 13. Okay. Khushbu, can you explain why you think that it is chromosome 13? You can unmute your mic. No, it's fine. If you want to answer, you can unmute your mic also. That why you think that is chromosome 13? You can write up also. You can open your mic also. Then we will move ahead to the next slide. And I will also explain you few of the very important things related. Okay, ma'am, because it's transferred. Fine, that is fine. This chromosome 13 is also transferring the genetic material, and this chromosome 21, that is also a chromosome that is also transferring the genetic traits. So these both are the chromosome. Then why specifically this 13 chromosome? What different thing you saw in this particular chromosome? That is basically, very much you think that it is relatable to the promotion of the breast cancer. If you have any clue. Okay, fine. You can think about it. And until you think about it, I will be telling you, my dear students, this it hardly matters that how many billion bases of are present in your chromosomes. Okay, there's one also in difference of bases. Yeah, dear. Yeah, dear. In the difference of bases, it is 114 million bases and it is 15 million bases. And you can see there are so many things written out here, the different types of, cancers. This particular gene is responsible for so many types of cancer. Okay, chromosome 21 is responsible. Okay. Okay, Sadiq has replied that chromosome 21 is responsible. Okay, Sadiq, why do you think that chromosome 21 is responsible for this promotion of breast cancer? Do you have any explanation? You can write up in the chat box itself. Okay, I guess no explanation. So that, okay, there's one more. Okay, because it acts as a negative regulator. Okay, fine. So there's also there will be so many questions, because if you say that it will act as a negative regulator, then definitely there is also the other chromosome there that can also act as a negative regulator. Because when during this COVID time, what happened? If you see the genetic content of the COVID virus was having, very less basis in comparison to this one human gene. If you say this virus, because these are, this RNA containing viruses, and they are, much smaller than these bacteria. So that have definitely, very small genetic system, very much less basis in comparison to the human. But that particular one virus with the very, very less basis, that has caused the pandemic in the whole world. So sometimes these genetic buildup, these bases, these millions or trillions or thousands or hundreds of base pair do not make a difference when it is there to cause any type of, disease. If there is any virus or if there is any bacteria that is a very small buildup of bases or a, very less number of genetic content, but if it has that pathogenicity, if it has some gene that is, 100% responsible for causing, highly exotic disease, then definitely those number of bases will not count. That will definitely cause, havoc and lethal disease in humans or animals or whatever, disease it is designated for, or whatever is the, host for that particular virus or bacteria. So, you can see here it is a 140 million bases, this chromosome. Definitely it has more number of bases in comparison to, this 21 chromosome that has only a 15 million bases. But if you can see, these have the specifications, this 114 million bases, and more than that, only the specifications you see. We have this dysplasia, muscular dystrophy, breast cancer, early onset, pancreatic cancer. Then it is also deficiency of MHC class 2 deficiency. Then it also this hypothermia. Okay, Aarti has answered that when I look at chromosome 21, the most possible disease is leukemia, Down syndrome, and breast cancer. Okay, that is a good thing that you're observing this. Fine. So, dear all, please mute your mics. I guess there is someone's mic is open. Fine. So there are a number of, bladder cancer, retinoblastoma, osteosarcoma, these, osteobone cancers, then your postaxial polydactyly type A2. And one thing very important, it has the MHC class 2 deficiency also. Then X-ray sensitivity, then Rigor syndrome, dementia, nocturnal enuresis, maturity onset diabetic condition is also there. Then pancreatic agencies. Yeah, definitely. Then coagulation factor X deficiency. So someone has answered that yes, in the chromosome 13, there are so many different types of cancer that can be caused by chromosome 13. Yes, definitely it is actually a very correct answer. Fine. There are so many different types of diseases that one particular chromosome is causing. So if this is present, definitely there can be n number of diseases. That person can have this X-ray sensitivity, then it can have the Rigor syndrome, it can have this coagulation factor also, and because of this coagulation factor 8 deficiency, there can be so many different other diseases. But when you see this chromosome 21, you can see there are also influenza resistance, myxovirus condition, leukemia, acute myeloid. Then you can see here also this is leukemia, transient of Down syndrome, then enterokinase deficiency is there, and mycobacterial infection is also there. Fine, that can also lead to mycobacterium species, for example, the tuberculosis. Then your epilepsy, syndromes, breast cancer is definitely written here. But in this condition, there are more number of cancers. You can see this osteosarcoma is a type of cancer. Then your bladder cancer is definitely a cancer. This retinoblastoma, that is an infection of eyes that that can lead to blindness, that can also turn into cancer. This pancreatic cancer, then you can see this cataract, if not treated timely, it can lead to complete blindness. Then stem cell leukemia. Stem cell leukemia is one of the very worst conditions for the patients. So there are so many things that can promote breast cancer. These diabetic conditions can also promote the breast cancer in any female, even in the males also. Although we generally think that these females have the breast cancer, but no, males also can the breast cancer, because although this physiology and this anatomy is a bit different, but definitely there is also the structural similarities between the male and the female, so they can also lead to breast cancer. Although these these milk glands are not present there, but that area can also get affected with the cancer due to many other causes. So in these both conditions, when you see in this chromosome 13, there are chances that the breast cancer can be promoted because of the certain types of cancers, secondary cancers. There are pancreatic cancer. This pancreatic cancer can lead to the breast cancer. This leukemia or B cell, this can also promote to breast cancer. This stem cell leukemia can definitely promote the cancers. The onset of diabetes in a very young age, that can also promote the this breast cancer because of those various metabolic disorders. Then your Rigor syndrome, that is very rigor mortis and all this is very different conditions where your bones get very much in a very contracted way, and it is very painful for also. Then your schizophrenia, that is a different disorder that comes under these psychological disorders. Then your coagulation factors, this Oguchi disease. So there are so many things that in this chromosome 13 that can definitely cause the promotion of the breast cancer. Now, when you see this chromosome 21, then again, there are few conditions that can lead to the breast cancer, but most of the these bases, these are responsible for certain types of autoimmune disorders and syndrome. For example, this long QT syndrome is there, this Down syndrome is there, your epilepsy, that is also genetic one, and some Usher syndrome, fine, chronic Usher syndrome, that is also a kind of disorder. So mostly these are the syndromes. These can lead to any genetic deformities. But when we talk about the breast cancer, we in the chromosome 13, we have certain different factors that can lead to the promotion of the breast cancer. Now, because we have not that much time to discuss on all these things very in a very broadly, so you can know this is example, and accordingly you can study at your home. You can do some homeworks on these things, that how a gene can be responsible for the promotion of breast cancer. So how many diseases, for how many diseases a particular gene or allele is responsible for. So it will be very interesting exercise for you to do. Then again, when we talk about the drug industries, there are so many drugs that are developed to combat the diseases affecting humans. Now, this is also a very important question, because pharmaceutical industry is also industry that is using this, biotechnology as a, one of the important tool. So most drugs are developed to combat diseases affecting humans, because when we talk about the drug industry, huge number of drugs, all the drugs are there only to, most of the, we can say 90% drugs are there, which are only, made to combat the diseases in the humans. If we talk about the animals, there are not so many drugs that are there that can, can be given specifically to the animals. But when we talk about the humans, there are so many drugs. You can see antibiotics are there for the humans itself. So many antibiotics are there. Fine. So you can see there is also a question, although we will not spend that much time on this questions. You can see here, there is a graph. This is the number of biotech drug candidates. That is the biotech, it is in the terms of pharmaceutical biotechnology and the industrial biotechnology in relevance with the drug industry. So there's also a question that which disease has the most drug candidates? Why does that disease have more drug candidates than hepatitis C? That means drug candidates, that means which disease has the more number of drug demand to cure that particular disease. So you can see here in this cancer, 465, according to this graph, we have 465 drug candidates for cancer. AIDS, we have 237. For the drug delivery ones, 117. For the infectious disease, there is 123. For the gene therapy, 75. Melanoma, 74. Vaccine, 73. And you can see the least number is of the colon cancer. But colon cancer is one of the very, very dangerous cancer that can lead to lethality also. But what we think that this HIV, generally what we say that AIDS and HIV, we don't have any cure. We don't have any cure, that is right. Once AIDS is there, definitely there will be, suppression of the immune system. Then why in this particular graph do you think that this hepatitis or these infectious disease, they have, less number of drug demands in comparison to the cancers? Anyone from this, audience team, can you answer that all diseases are very much, dangerous and they are lethal, but why the AIDS, your hepatitis C, your cardiovascular disease, your breast cancer, they have a very less drug demand? And that is actually the scenario. Do you have any, certain reasons why the drug demand for cancer is that much in comparison to the other diseases? Anyone from the participants? Can you answer this specific question? Whatever comes to your mind, you can, write up in the chat box. It will just give me, the thing that you are listening properly and you're attentive, and definitely you are just interested to know that what is the demand of biotechnology in the various fields. So any one of you, why you think that other disease have not that much of drug demand in comparison to the cancer? Okay, because in the cancer, the uncontrollable growth of cell occurs and it is very fast. Okay, Aarti is saying it is becoming more common in many people nowadays. Okay, that is okay. That is a definite thing. Okay, mostly cancers increase by rapid gene expression. Yes, that is also a good answer. Okay, anyone else other than Khushbu, Aarti, and Venkat? Anyone else? I will wait for, yeah, okay, mutations in cell. Any other answers? You can think, students, and you can write up, because this session is also for it's not only for you to listen and just finish up the session. It is also for you to think, because when we are talking about this industrial biotechnology, that does not mean that we are only listening and only going through all these things. Actually, when you think about something, that will make you think about the application of that particular thing. Okay, Srijan has answered that cancer has metastasis and angiogenesis. Okay, that is definitely a property of cancer. Anyone else? So my dear students, you are correct in a way, in a way or other way, you are definitely, about cancer, and it is correct. Why the cancers have more drug demands? Because, so in the AIDS, this AIDS disorder syndrome, basically what has happening, the suppression is there. Your immune system gets weak. Fine. And immune system gets weak, that means you can have a multiple disease. You can have cancer also. You can have any you are very much prone to any type of infectious disease. You can you can have any type of disease, even it's diabetes, it's influenza, your cardiovascular disease, your lung disease, anything can happen to you. But when we specifically talk about cancer, cancer is a very interesting and fascinating and a very we can say it is a diplomatic type of disease, because when we are suffering from any of the specific diseases, or it is hepatitis, or or you're having the cardiovascular disease, or you are having this leukemia is a type of cancer. So definitely mostly the drugs are not that much used in leukemia. There are so many stem cell transplants are there, and transfusions are there. So there is, I know, much better and quick alternative for these conditions. But in cancer, what happens is, because we have a group of uncontrollable cells, and then we are having the healthy cells, right? And these chemotherapies, these immunotherapies, these radiotherapies are definitely the cure for the cancers that are being used. These drugs are used, but these drugs, what they do, they also destroy the healthy cells. And because they destroy the healthy cells, when a person is, you must have seen the person who is going through a chemotherapy or a radiotherapy, there is a hair loss, all the body hairs, they get shed off, and then the person becomes very much weak, and there are so many blisters in the mouth, in the oropharyngeal tract, in the endotracheal tract, in the body, and there is so much of heat generated inside the body that a person will not die of the cancer, but he or she will die of the treatment that is given. That is the one important reason that why there is more drug candidates demand, that different drugs can be there. And the other thing is that when in the cancer we are eating the healthy food, we are eating healthy food. This is giving the abnormal cells the glucose, the energy to divide more. So that is why I said that this cancer is a kind of diplomatic disease. One side, we need ourselves to stay healthy during this cancer treatment. So that's why we are needing healthy food and healthy good vegetables and good antioxidants. But at the same times, these antioxidants and these healthy food, they are also providing the good nutrition to the uncontrollable cells. So they are also getting the good amount of nutrition. So it is, cancer is behaving like a very diplomatic one. Now, what to eat and what not to eat? If you will eat properly, then you will be, your cells, that cells will be, getting somehow, they will be getting good nutrition. And if you are going for the fasting, then definitely if you are not eating for too much time, definitely the cancer cells, they can, die of not getting the proper nutrition. But you can also die of not getting the proper nutrition, because you will feel weak and you are going through a very these high drug containing chemotherapies and radiotherapies. So that is why there is a no demand of drugs for this particular type cancers. All cancers are involved in this one, because treatment is there, but that has a worse side effect. If you are having the healthy diet, then that can also promote the growth of the uncontrollable cell. So there needs some mechanism, there needs some time of drug that can at least block somewhere this metabolism, where these uncontrollable cells can get the nutrition or any way. So that is why there is so much of research going on, because most of the people, they don't die of the cancer, but they die of the treatment that is being provided because of these harsh chemotherapies. They are very much painful and very much in many people leave the chemotherapies in the mid also. They prefer dying rather than getting the chemotherapy. So you after the session, you can go and you go and Google it, and you can study that how much worse is the chemotherapeutic treatment and how much painful it is for the cancer patients. Fine. So that is why we have the drug kind. Now, how you can relate this particular thing with your industrial biotechnology and the application. For instance, if anyone of you is interested in going in the drug or industries or pharmaceutical industries, you can think, or you are planning up for going in the research fields, you can think that what thing I can do, what drug I can make, what source I can utilize for the production of drugs that can actually aid in treating the cancers. So that will be the actual application of your knowledge into the industrial field. Fine. Now moving to the next slide. Again, now we come to the genetic industries. Now, what is biotechnology and what does it mean to you in the terms of when we talk about the genetically modified culture cells to make proteins of interest? Nowadays, so many, single cell protein, SSP, are utilized, and they are also available in the market. So that genetically modified culture cells to, make the proteins and to provide more nutrition and good amount of proteins, we are using this genetic modifications in industries. Fine. So you can see here in the picture, there's explanation that, for example, this is your gene of interest. This particular gene is, specified, or it has the, capability of protein or producing a good amount of, protein. Fine. So you can make a protein of yourself. You can take a gene of interest, introduce it into a bacterial or mammalian cell. That is, that is a genetic modification, or we can say also, we can sometimes we can call it also, it is type of recombinant technology also, because you are recombining a gene of interest, or you can inoculating your gene of interest, in a bacterial or mammalian cells. So you can see you're introducing this bacterial or mammalian cell into this culture plates. Now they will be growing with this gene of interest. Now these grown cells have the gene of interest. Now they are just transferred to the fermenter. Now it is not actually that much easy process as you are saying. So it is the way the flowchart looks like, it is not that much easy. It's just made easy for you to understand, right? So it's like the protein is not only the one that you are eating or you are consuming. Fine. That proteins are also available. That proteins can also be made with the help of this genetically modified cultured cell. The various vaccines or antibiotics that can also be made with the help of the particular one, that is this genetically modified cultured cells. So it just like that you are picking up a gene of interest, introducing it to a bacterial or mammalian cells. You are growing it on the culture plates, and that are not general the culture plates, or what you think like that we are just doing the certain microbiological practices. So it's not like just like that. It's special in a very specific conditions, in in vitro conditions. Everything is very sterilized conditions. All these preparations is done. Then the particular grown cells, and to identify and to cut out this gene of interest, the bioinformatical industries are required, the bioinformatical tools are required. Fine. So that is also one application of the bioinformatics in this particular genetic industries, right? So all these gene of interest, including proteins, antibiotics or medicines or your vaccines, are produced with the help of these techniques utilized in the genetic industries, right? So that is also one of the very important application. Then products of the modern biotechnology. Now, when we talk about this industrial modern biotechnology, what are the various products that we are getting? Now, if we talk about the examples of protein created by gene cloning, that we are mainly calling the recombinant proteins. These are the examples of protein you can see in this table. These are the examples of proteins manufactured from the cloned genes. Your blood factor 8 clotting factor to treat the hemophilia. So, when in the previous slide, where we were talking about the chromosomes 13 and 21. So we saw that in the chromosome 13, there was a condition where this blood factor 8 was there. That was, there, which it was mainly stopping the blood to clot. That was a condition. That was a disorder. Now, to treat that particular condition, we have the clone genes, we have the proteins that have been manufactured from the clone genes, and that condition is the hemophilia, and that particular disease is treated by that particular thing. Then epidermal growth factor, that is also a product, and the application is stimulate antibody production in patients with immune system disorders. So this epidermal growth factor, that is the product, that is a kind of recombinant protein. Fine. And what it is doing, it is stimulating the antibody production. That means if a person is immunosuppressed or immunocompromised, then what will happen? That particular epidermal growth factor, that will stimulate the production of the antibodies, so that it can fight back with the various antigens and infection. In that way, the immune system can be made strong. Fine. Then you can see there's the growth hormone, that corrects the pituitary deficiencies and short stature in humans. Then insulin. Okay, insulin, you must, it is a very, common thing. Can anyone tell me who was the first one to introduce insulin? Because it is one of the very important, it is like a, for the people having the diabetes, it is a boon to them. So anyone can tell me who introduced this insulin? Or okay, Venkat has said that isolates. Okay, anyone else? Anyone else, beta? Frederick Benting. Okay, is this that you're knowing it, or you are just going to the Google for the answers? Okay, anyone else? No, if you know it, that is a very good thing that you are actually knowing it. Okay, there's one different answer, that is Leonardo Thompson. And then Dipali is saying that it is Frederick Banting and his assistant Charles. Okay. Fine. Any other different answer? Okay, fine. So basically, every one of you is correct. You know why? Because the research, one very important thing about research is documenting the thing. Although this Frederick, he was there, and in the Toronto, he was studying, and I guess it was 90s time. I don't actually remember the year, but it was 90s, year 19 something, 1919 or 20, I guess. So he was there, and they introduced this insulin, because there was a no very much although that was the introduction of the insulin, but because of the for the proper treatment of the this diabetes and this therapy. Okay, there is, I guess, one more is answering. Okay, Eli Lilly is the first company. Fine, good. Commercialize the insulin. Okay, that is one of the very good answer. So, Aarti, I guess Aarti has answered this. That is a very good thing that that Eli Lilly is the first company who commercialized the insulin. And that is, one of the very important fact about the industrial biotech, that you can just think that a company that has commercialized the insulin and that was selling the insulin so that it can treat the patients of diabetes, then how much turnover for the company was there, and how much, we can say, how much humanitarian thing that company has done, because there are so many people. This COVID was a pandemic, but there was, if you see on the daily basis, this diabetes and cardiovascular diseases are the one of the most common diseases from which people die, on the daily basis. So because in the pandemic condition, there was a continuous deaths, and that was a recorded data. But on the daily basis, there are so many people dying of diabetes and cardiovascular diseases, and we don't have that much of data stored, because not everyone is visiting the hospitals or the government hospitals or the clinics or the laboratories. So there are so many people who are dying at the home itself because of the diabetes and these cardiovascular diseases, and without any treatment. So there is also a large amount of data that is not recorded in the government portals. Fine. So definitely these the companies who commercialize your any product, either it is insulin or it is any type of uh, antibiotic or vaccine, that is very much important, that who is actually commercializing it. Although they invented it in the previous years, they invented it, they introduced it, further the purifications were also done by the further scientists, but it came into existence, it came to be known by the people when it was commercialized, when people came to know about its market value, its medical value. Fine. So that is the importance, that how you can apply this biotechnology. And it's not like that you have to be very you should be getting very 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 high degree to get into this field. I will be giving you example of you must have heard about Himachal Pradesh. It's a state. So there's one person, I don't remember his name, but he is Bachelor of Arts, and he introduced this agriculture biotechnology in his area. And nowadays, in the present scenario, his exports of the hybrid and different types of plants that are in other countries, and in Kashmir also, he's just selling out his hybrid plants and potato and different types of ornamental fruits. Oh, sorry, this ornamental plants. And he's in the Himachal Pradesh. He's known as I guess his name was Deepak or something, Deepak Biotech. So people, he is not the degree of this biotechnology. He's just a Bachelor of Arts BA, but he's known to be this as Deepak Biotech, because what he was doing, he was not well known of the things. He went to the, because Himachal has also, there are agriculture industries are there, and this cloning, these genetic studies are done in the various universities there. So he used to visit there and ask people or ask the faculties that what is the problem and how we can do all these things, and he used to collect the knowledge. And that is why he's today known by anyone, and definitely he's very much good in the market. So it's not only about a degree, it's about the knowledge. My dear students, when you're studying everything, just connect it with yourself, right? So this answer of Aarti, I very much liked her answer, because it's a very good thing, although everyone was right. But when we talk about the commercialization, it is actually the one of the most important benefit and need of the current biotechnology, that how you introduce this thing into the fields. Fine. So these are various recombinant proteins, and these are the products: growth hormone, insulin, interferons, interleukins, definitely IL-6 and IL-1. These are important for the cancer treatments. Then monoclonal antibodies. So when you will be studying this genetics and recombinant DNA technology, these monoclonal antibodies will be there, and you will be very, very happy to see the mouse there. There will be a mouse and the diagram of a mouse, and the antibodies are prepared, and then they're introduced in the mouse. So that is just a visual a demonstration, but actually when we do it in the real world, it's a difficult thing to do, but definitely a very, very productive thing. So for the diagnostics and treating variety of diseases, arthritis is a autoimmune disorder, right? And in the current situations, there are so many people who are having this pain in joints and inflammations and loss of synovial fluids. Fine. And we are not known to these diseases. We know, we always, whenever we have a pain in our hands or legs or any joint, we just say, okay, I am having this arthritis, I am having this spondylitis. But actually all these diseases are autoimmune, and they have the same symptoms when they are on the very early stages. So it is very much difficult to distinguish that it is your rheumatoid arthritis, it is your reactive arthritis, it is your ankylosing spondylitis, right? So it is very much difficult. So gene-based therapies can be, you can study, you can go into the fields of gene-based diagnostic algorithms. The gene-based industries, it's on a topmost industries in at current time, where gene-based diagnosis is there. So there are so many autoimmune disorders for which it don't have any of the gene-based diagnostic algorithm. So you could go into the fields, then your tissue plasminogen activators, it is used to treat the heart attacks and heart strokes. So these are the various uses of the recombinant proteins. Then your types, now different types of biotechnology. That means all these are the various industries which are related to your biotechnology, your microbial biotechnology, agriculture biotechnology. My dear students, these are all the various fields where you can pursue your careers as a good entrepreneurs, also as a good scientist, also as professor, also as an inventor, also as a good startup fields also, right? So microbial biotechnology, your agriculture biotechnology, your animal biotechnology, your forensic biotechnology, and bioremediation, aquatic biotechnology, then medical biotechnology, regulatory biotechnology. There is endless terms related to biotechnology, your fermentation biotechnology. Then there will be your gene-based biotechnology. Everything is there in this biotechnology. Then when we discuss about this microbial biotechnology, mainly it is the manipulation of microorganisms such as yeast and bacteria. Now, this microbial industry, this microbial industry, it uses this microbial biotechnology. Fine. So, my dear students, can you please tell me in which, because I guess most of you are biotechnologists or microbiologists. I don't know because there are so many students. So, can you tell me which field of biotechnology, makes you like it's like, you plan for pursuing your career in that biotechnological field, or the field that you think that, okay, that attracts you very much. You can write it in the chat box so that I can know that which field is very fascinating for you, or the field which attracts you very much related to biotechnology. Which industry do you like? If you want to pursue your career in the future, which industry will you choose for your future? You can write up in the chat box so that I will be knowing that. Okay, medical biotechnology. Okay, that is my field also, Aarti. I'm also a medical biotechnologist. So yes, there is so much, career options in the medical biotechnology. Okay, anyone else? Okay, Nitin is also there. Microbial biotechnology. Okay, Niveda. So that means this microbial biotechnology, this particular slide is for Niveda, because she wants to pursue microbial biotechnology. Okay, Dipali, she wants to pursue biochemist. That is also a very good thing. You can, do great wonders in this drug industries, this pharmaceutical industries, because, this biology and this chemistry and this pharmaceutical, they all, when involved together, they can be, very for a great benefit for the people. Anyone else? So only 3 students wants to pursue their career, and others, rest of you, you're still thinking that which field will you join in future apart from medical, microbial and biochemist. So you have not that much time to think, but you can at least plan for yourself. Okay, agricultural, Nitin, that's a good industry, one of the major, industry, other than your vaccines and your genetic industries and molecular industries, the food that we need on the daily basis. So Nitin will definitely do great in agriculture industries and do good, invent, or we can say produce good hybrids or selective breeds for us, so that we can have a food for lifelong and for the coming generations. That's a good thing. Okay, now coming back to the slides, microbial biotechnology. So basically it's the manipulation of microorganisms such as yeast and bacteria. Fine. Creating the better enzymes, microbial enzymes. It is, there are endless number of microflora and fauna in whole world, and we can make endless number of enzymes. So we need not to make the enzymes out of we are not just not like that. We have to do day and night, and we using all the such sources which will be that will are not renewable. It's better to go for the microorganisms. There are so many microorganisms, and they don't need so much of it's not like that we are for us we need specifically AC or so many facilities. They don't need that much of facilities. You will provide only the limited sources to them that is required for them. They will grow and they will multiply, and you can prepare the enzymes from them. And definitely these microbial enzymes, they are produced and used in your dairy industries, your fermentation industries, your clothing industries. Fine, your genetic industries also. There are so many industries where we are using this microbial enzymes. Then more efficient decontamination process for industrial waste removal, for the bioremediation, for the wastewater clearance and decontamination. These microbial enzymes and microbes can be used. Fine, there are so many researches that are going on, and one of the very good research that is going on is the eradicating, or we can see decomposing of the plastic, because plastic is it has got the it has got the very good blessings from the God that it will live lifelong. It will be having very long life. So for reducing the life of the plastic, there are scientists who are working on the microbial enzymes that can actually decompose the microbes. Then your cloning and producing large amount of important proteins. These enzymes are used in biotech industries, for the medical biotech also, for the production of drugs also, fine, vaccines also, for using as cofactors. So microbial enzyme have a very much of benefit, and this microbiotechnology is an industry that will lead you for your dream work if you are interested in the microbial enzymes and microorganisms. Then agriculture biotechnology, for as ag food, that definitely there is, ultimately I am continuously talking about the food, that is one of the most important requirement for us. So this United Nations Food and Agriculture Organization predicts by 2050 we will need to feed a world population of 9.1 billion. So my dear students, you can imagine that by 2050 we need around 9.1 billion of people to feed. So Nitin Kumar, because you want to pursue agriculture biotechnology, so the future of the world is enhanced of people like you who want to pursue their careers in agriculture biotechnology, because yes, we need to eat, we need to have the food for living, for the survival. So there is a very much requirement for raising the food production. That means raising the food production by approximately 70%. That means it's a huge, raise up of the requirements. So feed up the population that is definitely growing and that is not going to reduce. So definitely we need the scientists, the people, the agronomists to work into the agricultural biotechnology industries to have some good alternatives for the production of the food. Then work in groups to brainstorm a few solutions to better feed the world by 2050. So that is why I said we need a group of brainstorm people. We need a group of good efficient scientists who can have a better solutions to feed the world by 2050. Then plants more environmentally friendly that yield more per acre. So that is what we need in the future. If you want to feed a large population, that means you need to have some plants that are very much environmental friendly, that yield most per acre, that can be done through the genetic engineering. Then resistance to disease and insects, that we also discussed earlier. Then foods with higher protein or vitamin content, that means a good nutritional fruits done with the selective breeding and recombinant DNA technology, gene technologies, that can be done. Then drug developed and grows as plant products. Then we have better plants ultimately reduce production cost to help feed the growing world population, because if there will be very much amount to be paid for the food, then definitely not everyone is going to have food, and still there are so many people who sleep without having the food. So definitely until 2015, I guess there will be a large population who will not be having a sufficient amount of food. So definitely there is so much to do in the field of agriculture biotechnology. Then, so basically, this is also a good task for you, that work in groups to discuss how you can use this technology in a third world country to create a better corn crop, main crop in that country. I guess most of you must have seen that movie, Interstellar, I guess. So you saw that what happened, they were going to the other world, to the space, and the only crop that was left in the world was the corn crop. So there was all the fields of corn, and there were so many storms that were coming, and there were so many diseases, they were destroying the corn crops. So it's there, you can sit up in a group of your friends and you can discuss that what technology can be introduced so that we can get a better crop and a better consumption food. Fine, because crop has all the 22 essential amino acids, because you need your brain, you need your mind to actually think about it. Because if you will think that okay, we will be having so much of wheat, we will be having so much of sugarcane, we will be having so much of pulses, then it is not going to work. You need to have an alternative. You can genetically modify a crop that will actually be having more number of nutritions, and that will require less area or less other resources, and that can be grown worldwide. That will not need any specific condition. That will grow, 12 months all year. It will be there. So that is the thing that you need to think as a biotechnologist when you will be working in the agriculture biotechnology. Fine. So will improved crops that are created to satisfy world hunger reduce available land for biofuel crops? You can discuss in groups. So I will again now continue with the animal biotechnology. So again, these animals are a source of medically valuable proteins, definitely. Fine. And antibodies, transgenic animals are there. Animals as important models in basic research that I also discussed earlier. There are hybrid goats who can give, nutritious and good amount of, milk. Your cows, hybrid cows that are also, healthy, fit and fine, and they can, give, they're also disease. Sometimes they are very much healthy and they can provide a good amount of, nutritious milk in comparison to the normal ones. Then so for the so many research, there are, animals that are used as the important models in basic research. Then animal cloning, source of transplant organs, that can be done. Animal cloning can be used for the transplant of the various organs. And, these vaccines and all, animal models are used for the vaccination purposes. Fine. So all these things that are very much, yeah, there. Okay, Aarti is saying there is a transgenic goats called the spider goats. Yes, that is, or there are so many different breeds. Definitely, and there are so many. If you are in this Indian Veterinary Research Institute (IVRI) in Bareilly, there are so many. They have the scientists are working on, because there is also, poultry farming and everything. There are fishes that are hybrided. So many different works are going on in this animal biotechnology. Fine. And definitely we have done so much of research, but now only we need to implement all these things in a much better way. Then we have the other industry, okay, transgenic animals. Fine, because all of you must be knowing that how the vaccines are made, and how we are in the older times. I guess we have a bit of time to discuss few things also. So I will take your another 10 minutes to discuss and wind up the session. So in the previous years, what happened, because there were some kings. Fine. So there was so much of chance that they will be, they will be getting these, poisons. Fine. So to tackle the situation, because what happened when the kings were given the food, the chef of the, the head chef tasted the food first, and then served to the king. So to ensure that there isn't any type of poison or there, because at the time there were not so many, just like we are having very safe home and all, there was not, because they had to go to the jungles and forest for the fights and all. So there were so many things that were done in that particular times. So at that time, what kings used to do, they were given a very, small amount of poison, or we can say small amount of any snake venom, in a very, very small amount. With the help of this snake venom, in a daily basis, what happened, the antibodies start to be, produced by the immune system. So they didn't die, but yes, they fall a bit of sick, but they get used to it, and day by day and time to time the dose of the venom was increased. You can study, you can go through the Google also sometimes, and you can go through such stories. So with day to day, the doses of the venom was increased, or the poison was increased, for which they were suspected that if, for example, if there was an area and there was cobra in that area more prevalent, so accordingly the poisons and venom was selected and they were given to the kings. So day by day, the strength of the king got stronger to resist the venom. So after one given span of time, if the venom, even if the king was bitten by the snake venom, that would not that would not cause any harm to the kings, and that was a way to keep safeguard the king. So that was kind of thing at the previous time, that was, our ancestors were very much intelligent. And now in the current time, when we are producing the vaccinations and this monoclonal antibodies, here also we are, but now today we are not using it on ourselves. Now we are using different animals and guinea pigs or your mice or animal models, horses, we are using them for as an animal model. So this is not, this animal biotechnology, very interesting thing, and you can definitely make your careers in this field. Now, okay, gene knockout, disrupt the gene in animal, and this is basically a task that can be given to you, but we have not that much of time. Then forensic biotechnology. Okay, I got one more answer from Aarti. Okay, she's saying, okay, from the milk of goats, they are extracting the spider silk thread. Yes, and the spider silk threads, that is, used for the many industries, for the clothing industries and all. And, silk saris and all, silk clothes are very much expensive, and that are, very, we can say, standard type of clothings they are considered. So definitely that is one way, that means we are not going for the silk worms. Now we are going to the goats to get that particular spider silk threads. So that is very interesting thing. Then forensic biotechnology, DNA fingerprinting, inclusion or exclusion of a person from subposition and paternity cases, identification of human remains. Nowadays in India only, there are so many cases of your rape and murders. On the daily basis, you must be hearing about there was a murder, there was a rape. So this DNA fingerprinting and forensic biotechnology, again, it has a very much demand, because now every time and with the year passing by, the pattern of murders are changing, the patterns of crime are changing, and for that particular thing, to take out the actual truth from these left over by the criminals, it is very much important to introduce new techniques, to introduce so many things. So in that field, your forensic biotechnology can help you out. And if you are the one who is interested in this particular field, definitely you will have a very great career if you pursue the field of your interest. Identification of human remains, endangered species, species, we are knowing, we are just from looking at the and counting the this tree, the stem of the tree, and counting the rings, we can identify the age of the tree, that what is how many years has gone uh, uh, to grow this particular tree. So these are also the kind of DNA fingerprinting. You can know with the carbon dating, you can know the age. You can also get to know the mummies that are buried in the ancient times. Fine, you can so get the age of that particular time, even if it's 100 years and 1,000 of years ago that was there. From the remains, from the carbon datings, we can get to know the age of that dead body or the remains. So all these things are very much fascinating. And in the forensic biotechnology, these alien studies that are related to the space sciences and all, that aliens ever existed, these all things to study the DNA. Because when I just recall one thing, I'm not that much I don't remember that much, but I was studying in a few days back. There is a species, a human species in Africa or somewhere, which has a very different DNA in comparison to the other people of the world. So that those people have a different traits and anything that is not that much similar to the DNA of all of the human. So that is considered as a foreign DNA also. So that is also one of the fascinating thing, that from which ancestry they must have been evolved. Was someday life was there on any other planet or not. So all these things are very good, and if you want to pursue your careers, then definitely this forensic industry is also a good option for you people. Then these are the based on DNA results from this gel, this defendant commit this crime or not. These are the gels. Fine, you can see here the gel pictures. For example, this is the sequence of one person and this presentation on the gel of other person, can we see that this was the person was, definitely the same or not? We can identify with the gel. That is called the DNA fingerprinting, where two different DNA samples are taken, the DNA is isolated, the reagents are prepared, and further your amplification is done for the specific housekeeping genes. For example, your human growth hormone, that is a housekeeping gene that is present in everyone. Fine. Or we can say these are the some genes that, if one person has the specific gene sequence, then definitely if that person has left his, leftover or any his imprints, that particular sample can be collected. And same, if the same DNA pattern is coming on the agarose electrophoresis or when visualized under the UV transilluminator or gel dock system, then it can be identified with the help of the DNA fingerprinting. In the same way, even in the micro field, even the forensic microfield also, if there was, for example, if in India or any other place there was an outbreak of a specific microorganism, fine, and that microorganism was found in, for example, in Australia, and not in any other places, that means someone was there in the India who must have visited to that particular place in Australia. That is why that specific species of microorganism was there, which was all in the starting was only the, we can say native of the this Indian country. So in that way, microflora can be also studied for so many forensic purposes and to identify any biological weapon. Then bioremediation, we already I also already in the previous one, I explained you about the oil spills, how we can use the bacteria to degrade the components in crude oil, where the oil spillage is there. In 1989, Exxon Valdez oil spill was there in Alaska. 200 and 2010 Deepwater Horizon spill was there. Because these, you can see in this picture, these are the oil spills and they accumulate, and these just prohibit and limit the this UV penetration into the oceans and seas, and that can lead to death of many these aquatic animals and plants. So this is what they are doing. They are just adding up the nutrients to stimulate growth of bacteria to clean up oil spillage. So in this way, they are just providing the nutrients to these areas so that the bacteria will take up the nutrition and they will grow, and they will just help out to degrade the oil spills. So this is just a picture. Then your aquatic biotechnology, your marine biotechnology, marine biotechnology is a good field, but only if you all want to pursue your career in marine biotechnology, that means definitely you must be around some, aqua, like sea or ocean, some place where near, because there are so many hidden things in the seas and oceans, that are the plants are there, and there can be so much of medicinal value containing plants, aquatic plants that are out of our reach, because we are not that much focusing on that particular thing. That may be definitely possible. So that is why this aquatic biotechnology is also an industry where aquaculture is studied, where raising the finfish or shellfish is control condition for us as a food sources. Then genetic engineering also is done in the oysters. Disease resistant strains of oysters are there. Mostly they're used for the consumptions, because of, if oyster is having some type of disease and someone consumes it, it can be very much lethal for us also. Then vaccine against viruses that infect salmon and other finfish to protect the various species of the this oceans. The vaccines are also there available. Then your bioprospecting, rich and valuable sources of new genes, proteins, and metabolic processes with important application for human benefit. Your marine plankton, zooplankton that you must have heard, and snails were found to be rich source of anti-tumor and anti-cancer molecules. So that is why exploring the seas and oceans, the aquatic life can be very much fascinating. And also in the terms of biotechnological industries, this also has a demand. But yes, definitely to study the seas and the oceans and the aquatic biotechnology, you must be there nearby the seas or oceans, because definitely that is the field you have to work. So that for you, that particular field will be your seas and oceans. You can see this is transgenic and fish. Two different salmons are there. One is, transgenic salmon, and one is the normal salmon. Fine. So how the modified salmon can help. So these are the animal models. Then medical biotechnology. It is involving the whole spectrum of human medicine, preventive medicine, your diagnosis of health and illness, treatment of human diseases, diagnostics, and so many different things are there, in this medical biotechnology. You can go for the disease diagnostic studies, you can go for the treatment of the disease, you can go for the cancer studies, you can go for the gene studies, you can go for the various study of various comorbidities related to various types of disease. So all these things, stem cell technologies, all things, they come under the medical biotechnology. Then stem cells that are on very much demand, which are grown in lab and then treated with different chemicals to allow them to develop into specific kind of tissues needed for transplant. The stem cell technology is on very much boom. Then these stem cells are definitely used for diabetes and spinal cord injuries. And when a baby is born, nowadays there are so many technologies where the umbilical cord is cryopreserved, because that contains the stem cell. So in future, if a baby is getting any such type of disease where the stem cell is required to treat, the mother also, even the mother also and the child also, that cryopreserved umbilical cord, there are cryopreservation centers, and they at the same time, as soon as the baby is given the birth, they store the cryopreserve the umbilical cord, and that cryopreserved umbilical cord can in future can help out the baby if the baby has got any type of cancer or any type of diabetes or any spinal cord injuries. And that umbilical cord, because this umbilical cord has provided the nutrition to the baby, and when the mother gets ill or any type of disease, through this umbilical cord only, the stem cells are provided to the mother to cure that specific condition. So this is a very interesting thing. You can work in groups to come up with a list of other diseases you have read about in the newspaper or heard on the news that scientists are testing with the stem cells. You can do that. Then there are so many things: gene-induced human obesity, new tests, use mother bloods, genetic switch puts hair on chest, and these are the news. And today you can go and study about the various uses of stem cell that are being done by the scientist. Then your regulatory biotechnology, quality assurance, quality control. If someone wants to be a biochemist and is going to pursue the career in the pharmaceutical industries, you can go for the quality assurance and quality control. That is one of the most important thing. Doing anything, either you are in an agricultural field, either in your pharmaceutical field, any field, without the quality assurance and quality control, you cannot pursue the work. You need the quality on its top to pursue anything. If you are using a reagent, for the gene cutting, or selecting a gene of interest and cutting it, digestion of that one, then definitely you should be knowing the quality of your particular reagent. Fine. So that is the importance of quality controls. You can go in the micro this research and development R&D sectors, as well as your policy QC pharma departments are there, QC biotech departments are there, your NABL, your ICMR, your WHO, your UNESCO, your UNICEF, as well as your NCDC, and so many different areas are there where the QC and QA people are required, and you can definitely pursue your careers on that. Then how will medical biotechnology change our lives in years ahead? Human Genome Project is there because research on the function of human genes and controlling factors. Human proteome is there, collection of proteins responsible for activity in human cells. Then your single nucleotide polymorphism is there, SNPs. Then, BRCA1, BRCA2, one of the most important genes involved in promoting the breast cancer. That is, that is a gene-based diagnosis of the breast cancer. So can you think of how this knowledge might be useful for someone who is not already diagnosed with cancer? So, dear students, I will end up the session with one of the very good question that I think, because this is the slide. Do you know any of the genes other than this BRCA1, BRCA1 and BRCA2 that are responsible for causing any type of cancers? Because, right now in the current scenario also, we are going for the biopsies, painful biopsies, taking a tissue section from a cancer patient and getting it tested that which type of cancer is this, going for so many invasive technologies to diagnose the cancer, going for the PT scans, giving the patient the contrast, giving the injections for the your scans and all. That is very much painful. But the gene-based technologies is very much useful. You can go for the non-invasive techniques. You can use the sputum, you can use the saliva, you can use the urine, you can use the blood, and you can just diagnose, okay, this gene is there. That means this person is having the cancer, this type of cancer. So do you know any of the gene that is, that, okay, this gene is responsible for this specific type of cancer, except the genes that are presented in the slide here? Anyone knows about any of the gene that is responsible for causing the cancer in humans or animals? Any of the gene, my dear students, that can be, helpful for the medical biotechnologist to diagnose any type of cancer. Okay, if you must not be knowing, there are so many genes, your XRCC1, XRCC2, XRCC3, A2A receptors. Fine, these are many type of genes that are responsible for the cancer in patients. And in the majority of cancers, you can go and study today your XRCC1, XRCC2, exon 6 and 5. You can go and study. These are some of the important genes that are related in causing the different types of cancers in the people. Fine. So gene-based technologies for the diagnosis of cancer and other diseases can be very good to at least save the people, because the patients are already suffering from so many of diseases and pain, and then they have to go for the non-invasive biopsies and all. That is, also that worsens the conditions. So why not to go for the, easier things, the gene-based diagnosis, very simple and easy, and that will not be, causing that much of pain and for the people who are suffering from very, lethal cancers. So definitely all these fields are very much important, and you can pursue your careers. These are the very broad industries that are there, which will aid you to start your companies, get the jobs in your various types of biotech companies, where you can have multiple degrees, where you can have, you should obtain a degree in your particular fields, your molecular biotech, your biochemistry, your bioinformatics. Then definitely maths courses are important. If you are planning to pursue the bioinformatics, then you should have a great skill in oral and written communication. You should be a collaborative team player. That means you should know how to work in a team. That is why you cannot work alone. You should learn to work in a team. Fine. And you should know to go into the field. That means that only does not mean that you have only to be in the lab and, working. You should go out and explore the world, and how the world is. Then only you will be, actually understanding the problem and the people. You should focus on your oral skills. You should focus on your written skills, your communications, because today you have to, be very much bold to have your firm legs in any of your fields. So to be firm and to be bold and to be, recognizable, You should be having a good overall written communication skill. Your teamwork will be highly appreciative. And there are so many. These are some objectives I will be showing you. ISO certifications are needed if you are starting up the laboratories. Then I will be giving you some overview of some instrumentations that are required in the various industries. See, this is the cooling centrifuge that is that is required in immunology industries and biochemistry industries and molecular industries. Your industrial biotechnology increase, these instruments are used. Your biosafety cabinet, that is one of the very important instrument that is setup that is much required in most of the industries. Your dry bath, your laminar airflow hood, fine. Then your multi-fuge spin wings that are required in most of the industries. That are the instrumentations that you should be knowing. Your PCR machine for the gene-based studies, your endpoint thermal cyclers that are very much important, your post-amplification machines, your agarose gel, where we were talking about DNA printing and all. So these agarose gel electrophoresis, very small unit, and it can actually tell the victim or who is the victim and who is the culprit. So this is only the machine, and you can tell about the culprits, and can find out who was the culprit and who was the victim. So then your ELISA reader for study of various antigen antibody reactions, your biochemistry analyzer, semi-auto analyzer machine, which can, you can study the various biomolecules in your body, your endocrinological system, all your thyroids, your cholesterol, lipid studies, everything you can do with the machine. Your cell counter, each and every constituent of your blood, you can study with the cell counter with the help of cell counters. So this is basically the clinical biochemistry and pathology laboratory. Each and everything you're in, either it's your plant tissue culture, either it's your plant pathogen based study, these instruments are very much required for you. BSL B2, then specifically designated separated areas. This is also, you must be knowing the basic difference between the laminar airflow and the biosafety cabinet, what type of filters are used. If you are working on the exotic microorganism, then what cabinet you will require. You will require a laminar airflow or biosafety cabinet. Then this is the PCR machine, again, very important for the medical biotechnologist, your pipettes, your all these cooling centrifuges, non-cooling centrifuges, your automation machine. So, if a manual process of isolating a DNA or RNA is taking 1.5 hour, this machine can give you result in 16 minutes. That is the upgradation of the technology. And the scientist is only a biotechnologist that has definitely worked in the engineering field as well as in the biotechnology field. So any one of you can go into such fields, and you can just come up with scientists or engineers that can make so many upgraded high throughput technologies for the study of the various fields of biotechnology. Your these electrophoresis unit, heating dry bath, so everything, all these instruments are very much required. Either you are working with plants, autoclave, one of the basic needed equipment for the microbiology students. Your blood roller mixture, how can you utilize the blood for the study. So each and every instrument that is required, how you have to store your reagents, that is important. Refrigerator, 4 degrees centigrade is required. For some need room temperature, some need 4 degrees, some need minus 20 degrees centigrade temperature, some need 80 degrees centigrade, minus 80 degrees centigrade refrigerator. So you must be, knowing each and every thing about this particular fields and studies, because this field is going to give you so much, and you can only learn when you will discuss all these things amongst your friends and your group of your batchmates. So thank you so much for attentively listening to the session. And if there are any queries, the platform is open, and you can definitely ask any queries. If you have any queries, I'm open to it. Thank you so much. If there are any queries, you can definitely write up in the chat box and I'm there to answer you. Anything, Bita. If you have any questions, Khushbu, if you have any queries related to the session, you can write in the chat box. You can ask in the chat box and I will be answering. If there are no questions, well and good. Thank you, Aarti. Thank you so much for this. And hopefully it will help you out for your future endeavors, this session. And I also thanks Make Intern Learn to Upgrade for giving me this opportunity to interact with the future of our country and dear students, because students are always very much important for us. Thank you, Amandeep. Thank you. So I guess there are no more queries. And thank you everyone for joining the session. And I will, give this particular stage to Learn to Upgrade. And ma'am, you can continue with the final proceedings. Thank you so much, everyone. Okay, I guess I should end up the session. Okay, I will end up the session. Thank you so much, everyone. Thank you so much.
Thank you so much, ma'am.